Capsaicin, a substance found in hot peppers, burns and even elicits pain on the eyes, skin, and mouth. But previous research has shown that there is no serious harm actually happening to the cells. So why do we experience pain, heat? Even weirder, how does it function to cause pain relief? Many have speculated that capsaicin mimics pain stimuli or activates the ligand released during injury. Studies have even shown that capsaicin's increase in cat ions excite pain receptors, but the underlying mechanism is still unclear. These speculations show that the discovery, research, and testing of the capsaicin target could lead to more efficient and effective treatments of all kinds of pain. The development and testing of capsaicin antagonist, capsaicin, and potent agonist re-inferritoxin, along with those dependents of these substances, has pointed to the activation of a specific receptor site to most likely be the mechanism of action occurring. Simply put, our hypothesis is that capsaicin's mechanism of action occurs through the binding on specific receptors in the neuron. Due to the lack of understanding of the capsaicin receptor's molecular structure, we decided that we need to isolate and clone DNA that interacts with capsaicin in vitro the same way it does in vivo. Based on studies resulting in capsaicin's ability to trigger robust calcium influxes into sensory neurons, we reasoned that complementary DNA, cDNA, of a capsaicin receptor that is conferred upon non-neuronal cells would also undergo an increase in free calcium when exposed to capsaicin. We used messenger RNA from the dorsal root ganglion of a rat to create a cDNA library, which are clones of vectors containing the RNA gene. The reason we derived mRNA from this ganglion is because stimuli that follow this afferent nociceptive neural pathway responds with a message that we interpret as pain. Due to capsaicin's responsive pain, this location is likely where the receptor responds to. The setup is as follows. We subdivided the library into pools containing around 16,000 clones each, which are vectors carrying the cloned gene. Each pool was temporarily transfected into HEK cells. Transfection is the process of moving foreign nucleic acid into the cell membrane via the vectors, and HEK is human embryonic kidney-derived cells, which are chosen due to their rapid reproduction and high acceptance towards foreign substances. So these transfected cells were then loaded with Furo2, a calcium-sensitive dye that becomes fluorescent when we expose the cells to a specific wavelength. We started with a control pool of HEK cells containing no vectors, so adding capsaicin did not result in a response. We then commenced the same process for each of our pools. Amongst all the pools, we noticed that pool 11, after the introduction of 3 micromolar of capsaicin, resulted in an influx of cytoplasmic calcium in around 1 to 5% of those cells. We then isolated this pool, subdivided the cells repetitiously until we isolated a single positive cell clone with a cDNA insert that was sensitive to capsaicin, then transfected that vector into a new line of HEK cells until we had a new line of capsaicin responsive cells. Since capsaicin specifically affects the vanilla type nociceptor receptor, aka temperature sensitive pain receptor, we deemed this name of this new cloned cDNA as VR1, or vanilloid receptor subtype 1.
We decided to call this Avalina receptor 1, the one we were able to harvest. Whether it has to do with temperature, and we know that capsaicin also responds to temperature. The next thing that we also decided to do is to make sure that this is capsaicin. This is a capsaicin receptor. For that, we also had to make sure it responded the same way as capsaicin does in the neuron cell. So we looked at the membrane potential. Then we added capsaicin to the cell. When we first started, we got this and added these receptors to them. And we did a whole voltage experiment and set it up so we could look at the membrane potential during stimulus. The eggs that now have receptors in it, we added capsaicin to them and saw a change. When we released it, it went back to normal, which was perfect because that meant that it was responding the way it needed to. Then after that, we added a strong antagonist and it caused change, but it didn't go back, so it killed the cell, which was also perfect because that's how we know that capsaicin was acting well. and it's responding as it should to what we already know. And then what we also had to make sure it was that it also responded to the antagonist. So when a capsaicin update, we also made sure it did the same thing. Then we waited a little bit. We added capsaicin with red rubidium, an antagonist that we know it works, or we knew it worked. And it did cause a change. So we knew it was responding the correct way. And then we also waited a little bit, and capsaicin, again, the receptor was responding correctly, and it was not being changed even after we added the agonist antagonist, except the strong agonist, which we already knew this was going to happen. Adding to that, we also know that it is dose dependent. We are looking at the properties of capsaicin on the screen right now and making sure these receptors do the same thing. So capsaicin is dose dependent, which means it gets more of it. If it gets more of it, then we get more response. As you can see, poblano, nothing happened because it didn't have any capsaicin, so it's responding as it's supposed to. And as we go up, we can also see a bigger change that correlates to how much response there is with capsaicin. We can tell that there's more capsaicin with habanero, and that is why it looks like that, as it should, which means we are getting close to finding that receptor that we are looking for.
We know that capsaicin is an excitatory neurotoxin that selectively destroys nociceptors. This is interesting because this actually brings pain relief. And that's kind of the whole point that we were working on this study. We wanted to know if capsaicin will kill the cells that have the vanilloid receptor 1. That was kind of our main thing that we wanted to know. And like we talked about earlier in the presentation, we know that capsaicin has the effect on the rats, and we wanted we wanted to know if it would have the same effect if we transplanted those cells from the rat to the humans to a non-neuronal cell that expressed VR1. So it's going to be in a different location in the body, because we already knew it was on the transplanted cell, but we wanted to know what it would happen if it was on a non-neuronal cell. And so after several hours of exposure to capsaicin, the cell actually died. And that was really interesting for us. And if you look on the y-axis, you can see the cell death on the side. And then on the x-axis, you can see our vanilloid receptor 1. And cell death increased dramatically, sometimes up to almost 75-80% when it expressed that receptor. So that was really, really interesting. And that kind of, proved our hypothesis. And the way that it would actually die after several exposures, the capsaicin would start to cause an extra influx of ions that would diffuse into the cell. And we learned about this in the first unit. And so it's been cool to see the comparisons. This would cause the cell to be hypertonic compared to the solution. And so we know when that happens, the cell will actually lyse, and the cell will die, and we will have more pain relief. And that's kind of the main thing that we wanted to discover, is the longer exposure to capsaicin means more cell death. More cell death means less pain. And that's simply put, but that's what we wanted really to find out. And so it was cool to make those comparisons.
Because capsaicin has a highly selective action, it is suggested that vanilloid receptors serve as specific markers for nociceptive neurons. Using northern blot analysis, we were able to visualize a 4 kilobase mRNA transcript expressed only in trigeminal and dorsal root sensor ganglia, both of which contain capsaicin-sensitive neurons. This transcript was not present in the spinal cord and the brain. A 1.5 kilobase transcript was detected in the kidney, but it is unclear if it could encode a functional VR1 protein. We were able to detect VR1 expression in a subset of sensory ganglion cells by using in situ hybridization. It is visible that positive staining in purple was limited to smaller diameter cells, which are pointed with arrows in both adult rat dorsal root ganglia at the top and trigeminal ganglia at the bottom, whereas large diameter cell bodies pointed with arrowheads did not express them. Two other tissues that have been proposed to express capsaicin receptors are the nodose ganglion, which contains cell bodies of visceral nociceptors, and the preoptic area of the hypothalamus involved in thermoregulation. By using in situ hybridization methods, we did not detect VR1 at either location, which could be explained by either a detection limitation from the assay or distinct VR subtypes, which has been proposed in other studies.
The burning quality of vandaloid-induced pain arose the question, is there a common pain molecular pathway for vandaloids and heat? We then explored the effects of elevated temperature on VR1. Initially, we recorded fluorescent calcium images of HEK293 cells during a sudden increase in ambient temperature from 22 degrees Celsius to roughly 45 degrees Celsius. In these conditions, cells exhibited only mild diffuse change in cytoplasmic free calcium. However, a large proportion of cells expressing VR1 showed a pronounced increase in calcium levels within seconds of heat treatment. The responses subsided in a matter of minutes, however, and produced a challenge with capsaicin, resulting in a characteristic calcium response. This suggested that the response to heat is a specific signaling event. To determine if a specific heat-evoked membrane currents are associated with this phenomenon, we examined VR1 expressing cells using patch clamp methods. We exposed the cells to a rapid increase in temperature from 22 degrees Celsius to roughly 48 degrees Celsius in 25 seconds, which produced a large inward current that was similar in amplitude to an application of capsaicin at 500 nanomoles. Both heat and vandaloid evoked responses showed outward rectification, which suggested that they are mediated by the same entity. These observations strongly support the hypothesis that VR1 is activated by noxious but not innocuous heat.
We also wanted to test how endogenous factors could affect our response as well. One factor we wanted to test was proton levels, or the amount of hydrogen atoms in our cells. The pH scale can help measure this value with a lower number representing higher amounts of hydrogen atoms present inside the cells. To start, we implanted our modified VR1 receptor into frog cells, which are then bathed in solutions of different pH levels. Our first test was ran by treating the egg cells of the frogs with a solution with a pH of 7.6, which was then dramatically reduced to a pH of 5.5. This test showed little activation of VR1 by the protons, so we wanted to see how lowering the pH and adding capsaicin would affect the response. This is shown in figure A on the left. In this test, we had a control group of cells injected with water and no capsaicin to show that no change would occur in the VR1 receptors. Next, we bathed the eggs in a solution containing a pH of 7.6 with the capsaicin until they reached a stable plateau. The current was then measured. Once the current was measured, we introduced the cells to a 6.3 pH solution, and we saw an inward current increase about 5 times greater than the initial solution. This is shown in figure B with a light gray column showing the cells in the first solution and the black column showing the current increase with the cells in the more acidic solution. After returning the solution back to an initial 7.6 pH, we saw the plateau go back to its original value. From this experiment, we saw alone that hydrogen ions were not sufficient to activate our VR1 receptor, but when paired with capsaicin, we saw a large capsaicin-invoked response, which makes us believe that the protons can actually increase capsaicin's potency of the VR1 receptor. In conclusion, we have shown through multiple processes that the mechanism of action for capsaicin is through a specific cellular receptor site. Along with its mechanism, we have shown how capsaicin works in a pathway and how it interacts with our body to elicit this response. Moving forward, we would like to use this information of the capsaicin pathway and apply it towards future research in pain remedies, such as analgesic medications. We're also excited for applications this pathway can hold in understanding how to treat conditions of acute and chronic pain. We are pleased with the results of our study and cannot wait for future applications.